You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.
I just had a quick question about your book. So are some of the other chapters written by other people, or is it all written by you, or what's the format of it?
No, there are 22 chapters. I write one of them. It's an enormous compilation of the exosome work in almost every disease you can think of, with obviously a huge focus on cancer, but on many other. You see, the fact is that exosomes communicate between cells and cells everywhere. So this has opened up an absolute can of worms. I mean, if you can communicate from anything to anything, including down to the germline, you've got a big problem. It opens up the number of possible interactions between cells in the body to an enormous degree. I think that's why the interest is so high. It can be a diagnostic tool, obviously. You can check whether you've got the profile of components, proteins, RNAs and DNAs that might indicate a cancer or might indicate some other disease. And all you've got to do is take a sample of exosomes from the blood plasma. It's in one sense, it's ridiculously easy, but in another sense, it's quite difficult because these things are extremely small. They're much smaller than a bacterium, about a fifth of the size of even the smallest bacteria, and so you can't see them with a light microscope. So there's no way of getting at them through the usual technique of just putting a bit of solution underneath a microscope. You've got to use really, really clever techniques to isolate and work out what's in them.
Well, how were exosomes first discovered, and who discovered them, and how?
That's a lovely question. You know, they've been known for ages. You see, I'm a physiologist, Rich, so I laugh at this kind of situation. But you see, long ago, maybe 80 years ago, we knew that nerve cells pour out chemicals in tiny vesicles. And what are exosomes? They're tiny vesicles filled with chemicals. The difference is that when neurophysiologists discovered that a nerve communicates with a muscle via acetylcholine, for example, we found that, of course, there were tiny vesicles liberated from the nerve ending that contain acetylcholine. The difference is that what exosomes, as they're referred to today, contain is bits of DNA, bits of RNA, bits of various proteins. In other words, a set of the characteristic of a particular cell in terms of its gene expression. And if that can in turn determine the gene expression of another cell, you've got a means of communication that is absolutely phenomenal.
So I think I see why this is so big. So what does this bring to mind for you? Let's say, like, within the heart, do you think that the different heart cells are communicating all the time, and that's how maybe they pacemake, or they perform other functions? Like, what does this imply?
No, I don't think that's necessary, actually. The pacemaker mechanism, if I can blow my own trumpet for a moment, was worked out a long time ago. It relies on electrophysiological properties of the ion channels in the pacemaker cells. We don't actually need this kind of cell-to-cell communication. All you need is a bit of electrical connection, and we know that that is provided by the what are called the goodness me, I just know the nexus junctions. That's right. That enable one cell to be electrically connected to another. So in that case, you don't actually need cell-to-cell communication via exosomes. But what's becoming clear is that most cells are not communicating electrically with other cells. What's becoming clear is that they're communicating, nevertheless, through the exosome route, which just means an extracellular vesicle, a bit of lipid globule, like a little bubble that contains a set of the molecules that come from that particular cell, which therefore can communicate the epigenetic effects to another cell.
Well, I have tons of questions about this. This is so amazing. So is there a organelle inside the cell that you think is responsible for creating the exosomes? And like, how does the cell know to create them, and how does it create them?
Yes, well, there is what's called the endoplasmic reticulum, which is another sort of fancy word for lipid membrane systems inside the cell. There are many of those, ribosomes and mitochondria and so on. But there is also a reticular network inside cells, and it's budding off from that network that you get the little bits, little vesicles that then can exit from the cell itself, just as a neurotransmitter exits from a nerve cell to communicate with another nerve cell or a muscle cell. So to answer your question earlier on, how long have we known about exosomes? As a physiologist, I would say for at least 80 years. What's different is this knowledge now that they can contain the epigenetic data. And that is phenomenal, because once you can communicate epigenetic data from one cell to another, and not necessarily as the same cell type, it could be down to the germline, for example, then you have the possibility of controlling the genome in those other cells. That's why it's so big.
Yeah, that's crazy. So what do you think is happening, for instance, in the human body? Are these cells of a particular organ communicating with other organs, or with the cells in the same organ, or with other body systems? Like, what kind of communication do you think is going on?
They're doing both, communicating to the cells within the same organ, but they're also, because they're poured out into the bloodstream, into the lymph vessels, and therefore into the body fluids in general, they're communicating everywhere. Now, what this book that I'm partly involved in, both in writing the chapter I wrote and in being one of the editors, is concerned with is precisely the question: to what extent can one use the measurement of exosomes and what they contain to give you diagnostic tools for anything ranging from possible cancer development through to why some males don't produce enough sperm to be fertile. I mean, the range is phenomenal. I've got in front of me, and I've asked the other editors whether I can send you at least the list of the titles of the chapter in the exosome book, and I'm just looking for it at the moment. I have somewhere here, can't find it just at the moment, a list, and it goes through virtually all the various disease states of the body.
Wow.
And there's a chapter on each of those. That's why the chapters number about 20.
Can you give an example of maybe the one chapter or the one disease that you're most familiar with, and talk about the mechanisms in which exosomes contribute to it or affect the disease?
Yes, where most people are focusing at the moment, because it gives the possibility that you can get a diagnosis for a cancerous development simply by taking a sample of exosomes from somebody's blood plasma. And that's what one of the chapters, of course, is concerned with. To what extent can this be used as a diagnostic tool to let you know whether cells are communicating what you might call cancerous signals to other cells, and therefore being part of the mechanism by which cancer spreads. I don't know, to be really honest with you, the extent to which that is actually going to work out in clinical practice. As I said earlier on, the detection of exosomes and being able to observe them is a very big technical problem because they're so tiny. As I said, they're smaller than bacteria. They're very tiny indeed. And so for them to be good diagnostic tools, people are going to have to develop the technology for detecting them and working out what is in them to a greater extent than is the case at the moment. And of course, some of the chapters I'm partly involved in editing are dealing with precisely that. How could the techniques be developed into good clinical practice?
Well, perhaps since the exosomes are being created within the cell, if you were to take a sample of a cell or two and then open it up somehow and see the exosomes before they left the cell, it might be easier to find out what they are. Maybe they take time to migrate out through the cytoplasm, out through the membrane. I don't know.
I see. Well, yes, the difficulty is there's so much else in the cell that would confuse you. I can only say, Rich, that what I've learned so far from reading some of the chapters that are coming in is that it's damn complicated. But I don't want to give the impression that it can't work. I think it can. I think it's like any new technology in biology. It'll need refinement to make it usable clinically, but I'm sure that will happen because the rewards for being able to do this properly are sufficiently great to make it worth doing.
Well, how, again, experimentally were exosomes observed? What equipment was required, and in what circumstance did it happen?
A very wide range of techniques are being used. Obviously, because they're so tiny, you have to go to electron microscopy to see them directly. So from that point of view, it's quite difficult because you can't be using electron microscopes on an ordinary clinical basis. So a variety of other techniques have developed. Some use what's called microfluidics, which is to detect within the blood plasma itself, the fluid, that once you've taken the cells out, you've got just the plasma, how you can detect whether microsomes are present. And the fact is that what you find is that wherever you take plasma, you'll find microsomes present. There are various techniques for using optical methods like flow cytometry, which is used to detect the exosomes in the suspension of the plasma. But the best comment I can make on this is that you probably need, and indeed the chapters in the book that I'm involved in editing show this, you probably need to use a variety of these techniques at the moment to get reasonable certainty that what you're looking at are indeed exosomes, in the sense that they are these little vesicles that communicate the pattern of RNAs and DNAs that come from particular cells. So I think the best answer I can give you at the moment is that you need a set of techniques to be reasonably certain that you've got what you're looking for.
And it would probably be a tall order then to say, well, okay, they found some of what's inside an exosome, but is there any structure inside the exosome? Is it like a mini cell or a mini organism, or is it just, I don't know, mRNA and things like that just sitting in this little vesicle, and it just, and also what happens to it when a cell takes it in? How does it take it in? Does it have certain receptors for it?
Okay. No particular difficulty with that. That's part of the story that we understand pretty well. Just as nerve cells secrete the vesicles that contain neurotransmitters, so cells can secrete through their plasma membranes the little vesicles that we call exosomes. And the mechanism is very straightforward because lipid membranes like to fuse with each other. So it's not difficult to see how, if you've got a vesicle inside the cell, it can first fuse with the cell membrane itself and then proceed to be expelled as a tiny vesicle by the cell. So we don't have too much difficulty understanding how it can be that the vesicles get out of a cell. As I said earlier on, as physiologists, we've known that for very many decades, that vesicles can be excreted from cells. As I said earlier, the excitement of the development of exosome technology today lies in the fact that they don't just contain an interesting transmitter like acetylcholine or noradrenaline or norepinephrine, but they can contain RNAs and DNAs. Now, to come to your question, how can the cell take that up and use it? We've actually known that from evolutionary studies on bacteria and other tiny cells, prokaryotes in general, for a long time. The bacteria are promiscuous. They exchange DNA with each other all the time. So there's no serious difficulty. Once an RNA can get inside a cell by being communicated through that exosome fusing with its cell membrane and then going inside, well, it's perfectly straightforward for it then to be incorporated into the genome, and we know that. There's evidence that is exactly what happens. You can show that even across different species, you can transmit DNA from one species to another through the exosome route. Now, the really exciting part of all of this, so far as I'm concerned, looking at it from the point of view of my interest in evolutionary biology, is that since that transmission can occur to the germline cells, we've been able to show, and that's what I show in my own article, that the exosomes can be not only transferred from one cell to another, but incorporated via the sperm into the egg cell to be then incorporated into the genome of the developing organism. And, you can't have something, it's hard to imagine something bigger than this in terms of epigenetic effects, because it means that you're actually changing the genome of an egg cell. And the implications of that are obviously huge.
Yeah, this is the missing piece to how acquired characteristics or epigenetic changes get passed down to generations.
Yes, that's right. I mean, that brings us back to Darwin's idea, and the extraordinary thing is how close the exosome work is to Darwin's original gemmules idea. See, he got onto this because he accepted Lamarck's idea that there had to be inheritance of acquired characteristics. There are around a dozen places in The Origin of Species where he says that. Now, later evolutionary biologists, particularly Weismann and Wallace, did not like this idea. They actually were almost embarrassed by the fact that Darwin had accepted some of Lamarck's ideas, and they tried to remove those aspects of Darwinism in what then became, of course, neo-Darwinism, the idea that really the genome was completely isolated. That was Weismann's barrier. The problem is that the exosome route via the sperm cells actually breaks the Weismann barrier. It goes straight through. In my own article, I refer to some of the work of Spadafora, who works in Rome, in Italy. And he's shown very clearly that the RNAs that can be communicated by the sperm can get incorporated into the egg cell, and that you then get it incorporated directly into the genome. So it's a transmission that is clearly communicating some information. The question is what information, but it's clearly communicating some information about the soma, about the rest of the organism that produced those exosomes, into the germline, which is the reason why I think that it's a possible route by which Lamarckian effects can occur. And that's what Darwin developed. After he wrote The Origin of Species, when he came on later to write about the domestication of animals and plants, for example, one of his later books, he elaborates this idea, which he calls gemmules, but they're exosomes. He says they are little particles that get communicated from one cell to another. He's almost giving the definition of an exosome. Yeah. Of course, he couldn't have known that the particles actually exist. It was to him just a hypothesis to try to reconcile his acceptance of Lamarck's ideas with his own work. But in effect, he was describing the exosome idea.
Well, I've heard that bacteria communicate, and they have things like quorum sensing, and that's when they decide, let's say, to infect a host, and they can count supposedly the number of the same, type of bacteria that are around and the type of other bacteria that are different from them. I know it's not species, but it's, it's another level. Do you think that, somatic cells can do the same thing and more? Do you think they are doing the same thing?
Yes, I think they're doing almost exactly what the bacteria are doing. They're, as it were, promiscuously exchanging DNA and RNAs. Yes, I think that's a good way to describe it. I mean, if you've got cells communicating with other cells everywhere in the body, and obviously initially in the same tissue, but then also communicating to other tissues, including the germline, then you've got a mechanism that's very much like the exchange of genetic material and RNAs in bacteria, but it's occurring, of course, in multicellular organisms. And that's partly why I think that from how should I best put this? From the fundamental aspects of biology, I think it's a major development, and it's certainly a major development given the implications for evolutionary biology.
Have there been any experiments you've seen where, let's say, someone takes, 2, 3, 5, 1,000 cells of a particular tissue, puts them in a petri dish, cultures them, and then tries to see if they're communicating and how and what exosomes are floating around in that dish? Is that possible?
No, I don't actually know whether that's been done. I'd be surprised if it has not been done. And I'm looking, as one of the editors of this book, as the chapters come in, for knowing whether a mechanism like that is described. Actually, in the titles of the chapters, and I have asked the other editors whether we could let you have the titles of the chapters so that you have a good indication of what the exosome book will be dealing with. And I think that would help you. And I think they'll agree. They're consulting amongst themselves and with the publisher, which is Elsevier, incidentally. But to come back to the question you're asking, have people done it in cell culture? I don't actually know. I've been watching as the chapters come in, and about half of them are already in. We would expect that the rest will be in within a month or two. I haven't yet seen that kind of experiment done, but I have seen a related experiment, and it interests me from the point of view of the heart, because at a conference I was present at last year in the south of France, in Sète, near Montpellier, some cardiac physiologists described a fascinating experiment. You can identify certain RNAs that will control the expression level of what is sometimes called the pacemaker current channel. That is one of the ion channels in pacemaker cells that controls the frequency with which the heart beats. And their experiment was lovely. It was to first identify some of the RNAs that are involved in controlling that particular protein channel, and therefore the extent to which it contributes to the frequency of the heartbeat, and then transfer those RNAs to the beating sinus node, that is the pacemaker region of the heart, of a totally different species. And the RNAs control the beating of that heart too. In other words, you can actually transfer the RNA information across from one species to another.
Wow.
So now that is it is tissue experiment in one sense, isn't it? Because after all, these are isolated tissues from the pacemaker region. I don't know whether that particular experiment has yet been published. It was presented at a conference that I was present at in September of last year, so just a few months ago. But I've no doubt that it will come out fairly soon. So you can transfer RNAs from one tissue, in this case the beating heart of one kind of one species, to another to enable it to respond by controlling its frequency. There's also evidence that the way in which athletes control their heart rate, you probably know that athletes can actually reduce their heart rate, which is extremely valuable in their performance, because if you can keep your heart rate low, there is a big reserve of cardiac contractile and frequency activity, which is what enables, of course, a really good athlete to have that kind of reserve in play to enable them to be better than, to be super athletes, as it were. Now, it was originally thought that the way in which athletes did this through their training was to, in some way, control the vagus nerve activity, because the vagus nerve slows the heart. And we've been able to show that that is not the way it's done. It's actually done epigenetically. It's done by various RNAs, whether in exosomes or just free RNAs, I'm not totally sure, being communicated to the pacemaker region of the heart of those athletes to slow the rhythm. And that's how the rhythm slowing occurs.
I want to, this is a question that's kind of, it should have been in the very beginning, but, I noticed that it's weird. It's so, I don't know why evolution and biology and these studies are so political. They shouldn't be. But what I noticed is it's weird. The neo-Darwinists, they don't seem to comment on things like you do. They get crazy. They scream and yell. They call you names. I've seen them call other people names. They laugh at people. Why do you think that happens with this?
Well, I think I can give you an idea, which I toy with myself, because don't know whether this is a correct comment or not, but I I notice this because I work sometimes in the United States. I go from time to time to do experiments for the Mayo Clinic. What I notice is that there's a bigger tension about these issues in the United States, and I think it's the clash with the fundamentalist religious people. My suggestion would be very simply that some of the neo-Darwinists, often themselves very opposed to religion. I don't have a position on this. I'm neutral with regard to whether people have a belief or not in religion. That's fine. It's up to them. It's their personal choice. But what does seem to me to be the case is that there's a huge number of people in the United States who seriously think that evolution never happened. That we are and have been developing much as Genesis describes it in a very literal way. Here in the United Kingdom, I think it's fair to say that the standard religions, I'm not talking about the sort of strange sects, but the standard religions, Anglican, Roman Catholic, Methodist, and so on, have long ago more or less accepted that evolution has happened, and that whatever you think about the religious implications of that, at least you're not terribly worried about it. And so the debate here is far less vituperative, I think. I think that's the best way to put it. And I think it might be largely a North American phenomenon.
Okay.
I don't know whether I'm right on that. That's my suggestion. Why it can get to be so heated, because it's heated on both sides. The fundamentalists amongst the religious believers are totally sure they're right, and of course the standard, if I can call them that, neo-Darwinist, if you wish, the evolutionary biologists are absolutely certain they're right too, and there's a clash there, a very clear one.
Well, you've been in science for a very long time. What does it feel like after 40, 50, 60 years in science, when you look around and knowing all you know, and seeing the complexity of life and the systems and interconnectedness? How do you sum it up in your mind? What does it make you feel or think?
It produces in me, Rich, a very deep sense of humility, because when I calculate, as I've done in some of my books recently, the total number of interactions that can occur, even if you take the simplest assumptions that there are only about 25,000 genes, actually we know there must be many more than that, because all the RNAs are essentially genes in the sense of being able to control and change the activity of the organism. But even if you take only the 20 or 25,000 identified protein coding genes, the total number of interactions that you can calculate from that is bigger by far than the total number of particles in the known universe. The known universe contains about 10 to the 80 particles, most of them, of course, protons, the nuclei of a hydrogen atom. And the total number of interactions that can occur between 25,000 genes is not 10 to the 80, it's about 10 to the 70,000.
Oh my God.
Absolutely enormous. This is in my book, so there's nothing strange about this. The calculation is very simple. You can do it with a bit of algebra and a bit of work on a calculator. Nobody challenges that fact. Now, it means, of course, amongst other things, that most of those don't actually exist. Most of those interactions can't possibly exist because there wouldn't be enough material in the whole known universe to enable them to exist. So evolution has obviously found a very tiny fraction of those that actually exist and actually work. But the point I'm coming to now, and the reason that this produces in me a sense of humility, which I have to say I didn't have 50 years ago. I was a good gung-ho, we're going to solve everything in the next 20 years. well, we didn't. and we're not going to solve it in the next 20 years either. It's going to take an enormous amount of patience and careful, what science is very good at, proceeding step by step to work things out, to really work out how that number of genes can interact to produce an organism. And it's been, I think, reasonably well proven now that this isn't a matter of imagining a single or just a very few genes being involved in a particular function, because what do the genome-wide association studies tell us? They tell us that you need to add together the contributions of a large number of genes to get a reasonable correlation with any particular phenotype, with any particular characteristic. Again, take athleticism as a good example. Instead of finding maybe a dozen or so genes that would give you an explanation of why somebody's a good athlete and others are not, you find it's hundreds, even thousands of genes. So that's even led some genome-wide association researchers to come to the conclusion that they call the Omnigenic Hypothesis, which is the idea that, well, practically every gene is associated with every function in the body in, minute contributions from each of them. What this tells me is that the original idea that there was a gene for this and a gene for that, and a gene, maybe just a few genes for this and that, it just isn't the right way to look at it. I see genes much more as sequences of DNA, as templates which the organism uses to enable it to produce the proteins it needs to function. And so the genome isn't so much an active causation in the organism, but a passive causation. It's used by the organism to enable it to create the proteins that it needs. Now, which proteins it needs, it'll be totally different for a bone cell, for a heart cell, for a liver cell. And this is where we come back to exosomes, because, you see, the exosomes from a bone cell and the exosomes from a liver cell and the exosomes from a heart cell will be totally different. They will contain the set of RNAs, DNAs, and proteins that come from that particular cell. That's why the information is so specific. We don't yet know how to understand all of that, because I think it is just so damn complicated. It's going to take us a long time to work it out. But what we can be certain of is that the information that is contained in those exosomes is cell-specific. And that, of course, is partly why when you come back to their role in disease and all the various disease states that will be described in the exosome book, the specificity is so important, because if from the exosome analysis you can get an idea of what a possible cancerous tissue is telling the rest of the body, then you've got cell-specific information on that cancerous state. And if, to take a healthy state now, you've got RNAs that come from a region that enable the heart rate to be controlled better, as in an athlete, you've got another set of expression levels of those RNAs and proteins and DNAs in those exosomes that is characteristic of a very different type of cell, a very healthy cell in this case.
Well, that's what I thought. I've talked to a number of cancer researchers, and I thought, well, why does cancer metastasize, for instance? And my theory, which has no support, but I figure the cancer cells are communicating with each other, and when conditions are right or wrong, they say, all right, let's spread. So that's how they know how to spread throughout the body. And with metastases, I wonder if the metastases communicate with the mother tumor, for instance, and what they're saying to each other. And I bet you they do.
Well, I'm pretty certain, Rich, that the chapter on cancer in the book that we're producing will address just precisely that kind of question, which is what is the role of exosomes in metastasis. I'm in a difficult situation here, of course, because I can't reveal to you what I know from reading the chapters that are coming in. What I have done is to approach the other editors and see whether it's possible that we can let you have for your website more details on what the book is going to contain, because I think that might help people.
Yeah, no, that would be great, and I'd definitely love to interview every single person in the book, too.
Yeah, I'll get an answer to that very soon. They're checking, and of course they'll have to check with Elsevier, the publisher, that information can be revealed. Yeah.
Right. Another implication that came to mind is, imagine now that we know a bit more about exosomes, how are the exosomes communicating? How are our somatic cells communicating with our microbiome, and vice versa? I mean, that opens up like a whole other world of craziness.
Yes, no, that is interesting. I agree. There's even evidence the other way. Microbiome DNA. I've forgotten now the exact article that I remember on this, but it concerns insect microbiome because there's evidence in certain insects that the microbiome has communicated with the DNA of those insects. I'd have to find that reference again. It's probably in my latest book, Dance to the Tune of Life. I'm pretty certain it's there. But the fact is that you're quite right. The communication with the microbiome both ways may be very important. You can be certain from what bacteria are capable of doing, which is that they communicate with each other all the time and exchange DNA. It's one of the reasons, of course, why they evolved so rapidly and are giving us a headache with regard to antibiotics, because they can transmit the basis of resistance to antibiotics between each other. And I've no doubt that they will be communicating with the host that they're living in, and as you say, possibly vice versa. At the moment, I haven't seen any clear evidence that exosomes, it's so easy to get these muddled up, isn't it? The microsomes, of course, are inside the cell. They become exosomes when they go out of the cell. I haven't seen any direct evidence that exosomes actually communicate with the microbiome. And that might be because there is, after all, a very strong barrier between the blood circulation and the lymph circulation and the gut. So it wouldn't be easy for the transmission to occur.
Well, what if this plays a role in the immune system? What if part of the way the immune system works is that it picks up on exosomes from, bacteria that bacteria may release, and it reads them and sees what's coming or what's in the environment, and vice versa?
Well, I have to say, Rich, that's a lovely thought. I hadn't thought of that myself. You're right, of course. After all, the immune system is consuming the bacteria. Yes, and if they can consume it, they can get the DNA.
Yeah,
yes, lovely. Yeah.
I don't know.
Might be true.
I guess I have so many questions. I feel like at a loss. I don't even know what to ask because there's so much material to talk about. It's crazy.
Well, to be honest, Rich, I'm in the same position because, you see, as I read the chapters that are coming in this book, I'm learning a huge amount of stuff that I never knew. And so I'm, after all, a practicing biologist and medical scientist. And I also feel, that the implications of all of this are huge. The question whether any of it is really practical and can be used clinically or used now to take an evolutionary perspective in the process of evolutionary change, those are very big questions indeed. And I think we're only at the beginning of addressing those questions, particularly in relation to the chapter that I wrote, which I think I sent you, which raises the very clear possibility that the transmission down through the germline can occur. And I'm going to be watching this area, of course, very, very closely indeed.
I've noticed by interviewing lots of people that everyone is kind of in their own world, and I haven't really yet seen someone that has let's take cancer, okay? I've never yet seen a mind map of all the possible implications of cancer, for instance, and all the things it could affect. And I've never seen anyone take a 10,000-foot look on everything that's going on, or almost everything that's going on within a field. I mean, even if you didn't delve into it, but you had this map in front of you, and you looked at it and kept it in mind, I would think it would help you in your work. Like, for instance, I've spoken to one lady that her whole research is about cancer metastases, and I asked her about cellular communication in cancer. She's like, no, they don't do that. And she's not even aware of this. Like, what do you think would be a better way, I guess, to do science and to use collaboration to benefit science? Because it doesn't seem like it's happening. It seems like it's happening in silos. It's weird.
Yes, you're absolutely right. The siloization of science is a problem, a very big problem. And of course it's exacerbated by the scrambling for funding, because you get funding largely by staying in your own silo. You don't easily get funding if you wander outside it. And that's a big difficulty. The reaction of a committee will be, well, he hasn't done anything in this area yet. Well, damn it, if you're proposing to do something new, you probably haven't done anything in the area before. It's going to take the courageous people who can, as it were, communicate across the divisions of the fields to really push things forward. I really don't know what the answer is, though, this is an organizational problem in science, isn't it? How do you encourage the interdisciplinarity that enables people to be courageous enough to enter another field? Because the evidence from the history of science, I think, is very clear. It's often the people who do the transfer from one field to another that produce the major advances. After all, it was physicists, essentially, Watson and Crick, who got the story of DNA essentially right and worked out. It wasn't biologists who did that. And you need this cross-fertilization. And how one then encourages that, given what you've identified, I think, correctly, as the silo mentality, I'm really not sure. The scramble for funding is producing a situation, and of course the tendency of universities to measure your scores and citation indices and so on is producing a situation where it's much easier to stay in the same narrow field than to wander around.
Yeah, they should say, hey, cells are communicating constantly. How come you're not?
Yes, indeed. That's right. This is a kind of cultural exosome problem, isn't it? How do you enable the transmission to occur? And people like me, of course, are in an enormous privileged position, because I've been retired from my original chair in the University of Oxford now for nearly 15 years. And I've been able to indulge myself in all kinds of activities that I wouldn't have dreamt of doing while I was responsible for a team of 20 people and trying to get their salaries all the time. When you're dealing with that, you really will focus on getting those salaries because you're responsible for them. So, there is a tendency for science, as you said earlier on, to stay within its silos, and it takes a few brave souls to go outside that. Those who are not in a position where they have to carry on doing that kind of treadmill, like me, tend to be much more adventurous, I think. There should be more of us.
So how do you do science today versus the way you did it, 15, 20, 30, 40 years ago? And
well,
what lessons have you learned? What's new with you that you're working with?
Yes, I've actually branched out instead. I mean, I, as you will probably know, I mean, my major contributions in physiology were to the electrophysiology of the heart. And for something like 45 years, I was totally focused on the ion channels and other transporters in the heart that generate its electrical activity. What am I working on at the moment? I'll tell you. It's a medication that was developed and described in a publication that has as its date AD 200.
As what?
It's the Shang Han Lun, which is one of the earliest volumes describing Chinese medicine. And I've been working using a variety of techniques and a variety of computational methods too to work out why that particular medication works. These, of course, are herbal extracts. So I've jumped away from my standard field of cardiac electrophysiology to use the same kind of ideas, that is a systems approach to biological problems, to try to analyze a medication that's been known for over 2,000 years. So that's what I've done.
Okay.
But I can do that because I'm no longer on the treadmill and in the silo.
Right. Are you surprised that medicines work at all, seeing the complexity of life and the complexity of all these systems?
Indeed. And you see, that also explains something else which I think is very worrying. If you look at the graph of successful launching of new medications against time over the last 60 years, it is a relentless decline in terms of the number of new drugs produced by the pharmaceutical industry against the fact that the investment to produce each of those new drugs is simply mushroomed. We're now in a situation in which it requires research that costs you of the order of $1 billion or $2 billion or even $3 billion to have a reasonable chance of getting a new drug onto the market. That's the scale of the problem. And it's nowhere more illustrated than the scare over antibiotics, where largely the pharmaceutical industry has not met the challenge, which is to find as many new antibiotics as we can, given that the bacteria are evolving fast enough to make many of our defenses be fragile, to say the least, and possibly even useless. We'll have to wait and see, of course, how the situation develops.
Do you think that medicine has discovered the low-hanging fruit, and that's why it's so much harder to find them?
Absolutely. I think that's exactly what happened. And, of course, the low-hanging fruit started with antibiotics. That was a fantastic discovery, penicillin and later ones. And, of course, if you're interested simply in killing bacteria, then, of course, you've got the right kind of approach. And, of course, the what you call the low-hanging fruit of being able to identify some of the key receptors that hold the main physiological functions of the body, drugs have been found to deal with those. But when you come to the complex diseases of modern society, particularly aging society, diabetes, the problem of obesity, the problem of very complex interactions of cardiovascular disease and immune system disease, what you find, of course, is that there's no longer a single drug that will do the trick. And what you find, you go around, it must be true in the United States as well as it is here. If you go around a general ward or a cardiovascular ward of many of them very aged people being treated, you'll find that the medication nurse goes around with a cocktail of maybe 6 or more drugs per patient, which are being prescribed because no single drug will do the trick. Now that brings me back to, if you like, the wisdom of some of the Oriental medication. They are, of course, multiple. They're not single chemicals.
Right, and because they come from an organism, so
they come from organisms that have developed over billions of years of evolution. And after all, why do plants have particular chemicals in them rather than others? I mean, there's only a minute fraction of the total range of chemicals that there could be that you find in plants. It is because they have function. The plant either needs those chemicals itself, or it needs those chemicals to defend itself, or it needs those chemicals to be attractive to other organisms that eat it and distribute its seeds, and so on and so forth. So from an evolutionary perspective, it's reasonably understandable why most plants will produce a range of chemicals that have functional effects, even in combination, because that's exactly what they've been evolved to do.
I guess also for purposes of redundancy, like, I had seen in your writing when you talked about the heart, and you had, it has redundancy and to ensure that it pacemakes, it beats, and does what it does. So
absolutely so. Yeah. No, you've got that absolutely right. I showed around 30 years ago that the pacemaker is very redundant. You can knock out one of the key protein mechanisms, or its gene, that is responsible for cardiac rhythm, and you get very little change in frequency. It doesn't mean to say that that particular protein is not functional. On the contrary, we know it is. But what it tells you is that there's enough redundancy there to enable the heart to go on beating away even when one of its major mechanisms is knocked out. And I think this is general. There was a study in yeast about 12 years ago by Hillenmeyer and his colleagues. They did a systematic knockout, one by one, of most of the 6,000 genes in yeast. 80% of the knockouts don't seem to produce a phenotypic effect in terms of the reproduction and metabolism of the yeast. And again, that doesn't mean to say that they're not functional. It's simply that the yeast has so many different ways of coping when it lacks particular proteins that it can manage, and manage pretty well. And I think that's fairly general. It's certainly true in relation to the heartbeat that you've got several mechanisms, any one of which can make sure that it continues.
Well, so if you think about the fact that a lot of life seems to have redundant systems, and I'm sure this extends to bacteria, and you look at the ability for these organisms and us to evolve in response to pressures and, adverse effects, there's no wonder that a single medicine would be lame against a creature that can do that and has those redundancies.
Yeah, that's right. Yes, that's absolutely right. Now, what we do about this from the point of view of dealing with the challenge of modern medicine in relation to aging populations, that's one of the huge issues that we have to face, and I'm not sure what the answer is. As I watch which I had to do when my wife was in hospital for several years before she passed away, I watched the way in which doctors treating a complex of interactive disease states were reduced to having what I called a cocktail of 6, 7, 8, or even more drugs being prescribed to the patient. Now, has anybody done a clinical trial of all of that? Of course they haven't. You can't do clinical trials of multiple medications at once. It's difficult enough to do clinical trials with just one medication and a placebo. So we're already actually feeling our way with multiple medications being prescribed to deal with multiple disease states, and that's the best that people can do. But we've no idea what the interactions are between many of those drugs that are prescribed together, because they, as I said, you can't do many clinical trials with multiple drugs. There's just too many combinations to make that financially possible.
Hmm.
So there's a big problem there, which with some kind of it's a technological problem in one sense, of course, but it's also a conceptual problem. Part of the reason why I go back to the idea that the plants have developed combinations of chemicals that are effective is that they've done it for us. They've taken billions of years to do it, but they've done it.
Yeah.
And of course, it's been the evolutionary process that has enabled that to happen. We're not going to find that within just a decade or two, we're going to be able to reproduce all of that. But I think that's a reason why I would recommend much more attention should be given to plants. As indeed it was, 200 or 300 years ago in the first and early developments of Western medication. After all, many of the drugs in the Western pharmacopoeia actually came from plants in the first place. They may now be made chemically, of course, as a chemical synthesis, but that's where often they first arose. And I think it's probably around 50% of the Western pharmacopoeia that has as its original basis the presence in plants. But that's why I'm doing what I'm doing, you see. That's why my present interest has switched. I'm leaving the heart to those who can be much cleverer than me, pushing the heart forward and moving into another area that very few other people seem in the West to want to do, but which I'm finding very rich and rewarding.
Okay. Well, I know we've been on for a while, so I want to be respectful of your time, so we'll finish soon.
Sure.
If you would, can I ask you one more question? And it's probably a big one, but all the talk about evolution, that I've heard is it takes, I don't know, thousands of generations and millions of years, or, at least many, many, many years. Have we ever observed the creation of a new species or of such a drastic change in an organism in one generation that we can see evolution at work? Or does it always seem to happen over, a long period of time and we're never able to visualize, see when it happened?
That's a lovely question. I'll give you one answer. There is, this comes from the Galapagos Islands, which of course has a huge iconic significance in evolutionary biology because it's where Darwin got his ideas in the Voyage of the Beagle, of course. Now, those islands are pretty far flung from each other. Well, not all of them, but some of them are pretty far flung. There can be as much as 100 kilometers between one island and another. A single finch from one island, a male, got across that long sea journey to a different island, and it bred with the finches that are a different species on the island that it got to. That's produced a new strain. I can get you the reference for that if it interests you, and I think that one is also quoted in my recent book, Dance to the Tune of Life, but if it isn't, it's certainly quoted in one or other of my recent articles. It's what James Shapiro would call the effect of hybridization, because after all that produces a hybrid between one species and another. And in effect, within a single generation, that has produced a new strain. Now, whether you call it a new species, I mean, that's a big question, of course. Definition of a species is problematic. Usually it's whether the animals can breed with each other. But even that is not a secure definition of a species. But that's perhaps the most dramatic example because it's well documented. That was a paper I think in Nature that I'd be able to find if you really want it. But it's very rare. You're quite right. Generally, evolution will occur so slowly that you can't observe it in just a generation or so.
Well, that's why does it occur? If it occurs slowly, it seems to Why does it occur slowly? Why can't it occur in one generation? And there must be a tallying of changes that build up and build up and build up, and all of a sudden there's a tipping point. Because otherwise, how could it happen like that?
Yes, I think the tipping point idea is a very good one. I remember a debate that I chaired about 10 years ago between Richard Dawkins and Lynn Margulis. Lynn Margulis was the great proponent of symbiogenesis, and Richard, of course, was defending the standard neo-Darwinist evolutionary biological viewpoint. It was a very interesting debate, which it's recorded too and still available. And what I found was that how best to put this, I was chairing the debate, of course, which is not a good position to be in. You can't sort of weigh in yourself in the way that you would love to do. But it seemed to me that, they came to the evolutionary process from very different perspectives. To come to your point, Lynn, of course, she thought, because she passed away just a few years ago, not too many years after that debate, but she thinks that symbiogenesis is essentially a single generation phenomenon, and the way in which bacteria got absorbed into other prokaryotes to produce what eventually became mitochondria was that they were eaten, but not just eaten by the other prokaryotes, but they managed to survive inside the organism that took them in. So they were ingested, but instead of being digested, they were used. And, of course, they became the energy-producing factories of all the eukaryotic cells that we have today, and we wouldn't exist if we didn't have those mitochondrial energy-producing parts of our body. All of them developed from what Lynn would describe as a single-generation acquisition of one organism by another. And so certainly Lynn Margulis would have said evolution can go in single jumps.
Okay. Yeah, I just wondered about that.
Yeah, well, there's my answer. And
very good.
He was a fantastic but cantankerous scientist, I have to tell you.
I guess you need to be,
She wasn't as good a debater as Richard Dawkins. Richard is an extremely good debater. Yeah.
Well, very good. So for listeners, what does the book have a name, the Exosome book, and when is it going to be coming out?
Yes, I don't know the exact date of its appearance. I think it'll be during 2019. But as I said, I will, as soon as I get a reply from my co-editors, I'll be able to let you have more details of it. But it's simply called Exosomes in Disease and Health. I think that's the title. I'm trying to find it here. Yes, I'm just getting it up now. Although that's the title of the book, Exosomes in Health and Disease.
Okay. And hopefully it'll be as exciting to people as a new Harry Potter book, because it's got such amazing info. So, Dennis, can you hear me? Did I lose you?
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.